Hello students, welcome back to my channel. In this video, I am going to give you objective type of questions from the unit 4. The topic is immunological techniques such as immunoblotting and ELISA. Let's see the questions one by one. Western blotting is the technique for the detection of. Options are DNA, RNA, proteins, lipids. So, Western blotting is a technique, it's a blotting technique that is used to detect the proteins. Let's see the next question. PCR technique was developed by. Options are James Watson, Carey Mullis, Francis Crick, Rosalind Franklin. So, the correct answer is Carey Mullis. He developed the PCR technique. Let's see the next question. So, which of the following techniques is used in DNA fingerprinting? Options are Western blotting, Northern blotting, Southern blotting, ELISA. So, for the DNA detection, Southern blotting is used. So, correct answer is Southern blotting. Let's see the next question. What is the purpose of PCR technique? Options are amplification of protein, amplification of RNA, amplification of DNA, none. Yes. So here the DNA is amplified by using a technique PCR. Let's see the next question. What does ELISA stands for? Options are enzyme linked immunosorbent assay, enzyme linked immunoassay, enhanced lymphocyte immunosorbent assay, enzyme linked immunosensitivity assay. So for this, the correct answer is enzyme linked immunosorbent assay is the correct one. So let's see the next question. Identify the thermostable polymerase from the following. Options are DNA polymerase 1, Taq polymerase, RNA polymerase, reverse transcriptase. For this, the correct answer is Taq polymerase is a thermostable polymerase which is isolated from Thermus aquaticus bacteria. Let's see the next question. Which of the following steps involved in PCR in the correct answer? So let's see. So PCR consists actually three steps. There, first one is denaturation, second is annealing, third is extension. So the correct one is here. B is the correct one. Let's see the next question. What is the purpose of blotting technique? So here the options given are to separate molecules based on their size and charge, to detect and analyze nucleic acids or proteins in the sample, to transfer molecules from a membrane to gel, both A and B. For this the correct answer is yes, A and B are the correct one. Let's see the next question. Which is not application of ELISA. Options are detection of hepatitis B markers in the serum, percentage of HB in the blood, detection of HIV antibodies in the blood sample, detection of mycobacterium antibodies in tuberculosis. For this, the correct answer is percentage of HB in the blood, which is not an application of ELISA. Let's see the next question. A researcher wants to detect the specific antibody in the sample. As a researcher, which type of ELISA method do you want to suggest among all the types of ELISA? Options are direct ELISA, indirect ELISA, competitive ELISA, none of the above. For this, the correct answer is indirect ELISA. Okay. So let's see the next question. Taq polymerase used in PCR is extracted from. Options are Escherichia coli, Thermus aquaticus, Saccharomyces cerevisiae, Bacillus subtilis. As I told you earlier, it is obtained from Thermus aquaticus. Let's see the next question. What kind of transfer is performed in Western blotting technique? Options are diffusion, magnetic transfer, electrophoretic transfer, none of the above. For this, the correct answer is electrophoretic transfer. In the Western blotting, so by using electrophoretic transfer, we can separate the protein fragments. Okay, let's see the next question. Which component is commonly used as a detection method in an ELISA assay? Options are radioactive isotopes, fluorescent dyes, enzymes, magnetic particles. For this, the correct answer is enzymes is the correct answer. Let's see the next question. So, in gene therapy, the gene defects are cured in dash stage of life. Options are adult stage, teenage, old age, embryo. For this, the correct answer is embryo is the correct one. Let's see the next question. The genetic defect is corrected by options are gene therapy by the delivery of normal gene into the individual, stem cell therapy by the delivery of mutant gene, therapy by the delivery of incorrect gene into the individual, RNA interference. For this, the correct answer is gene therapy by the delivery of normal gene into the individual. Let's see the next question. The direct ELISA is used for options are detecting antigen, detecting antibodies, detecting enzymes, detecting DNA. For this, the correct answer is for it is used to detect the antibodies. Let's see the next question. ELISA allows for rapid screening and quantification of presence of dash in a sample. Options are amino acid, DNA, antigen, protein. For this, the correct answer is antigen. Is the correct answer. Let's see the next question. Arrange the following steps about ELISA in correct order. Options are incubate with antibody enzyme complex that binds primary antibody, ward surface with antigen block unoccupied sites with a non-specific protein, add substrate formation of colored product, increased presence of specific antigen, incubate with a primary antibody against specific antigen. For this, the correct answer is okay. So this is the correct answer. That means second one is the first, then fourth one, and then first one, and then third one. So that is the correct sequence. So let's see the next category of questions. They are nothing but fill in the blank type of questions. Let's see the first one. The enzyme used in PCR to synthesize new DNA strands is called. So, for PCR, there is a special polymerase enzyme is used, that is nothing but Taq polymerase. Let's see the next question. What is the full form of ELISA? As I told you earlier, enzyme linked immunosorbent assay is the correct one. So let's see the next question. ELISA can be used to detect the presence of dash in a sample such as proteins, peptides, or hormones. So for this, the correct answer is antigens in the sample. Let's see the next question. Dash enzyme is commonly used in ELISA to produce measurable signal. So here the correct answer is horseradish peroxidase, which is commonly used in ELISA. Let's see the next question. Probe is. So here you have to define the probe. Probe is a short piece of labeled DNA or RNA which are complementary to the nucleic acid strand to be detected. Okay. So let's see the next question. Next category of questions belonging to very short answer type questions. What is the principle of ELISA? So ELISA enzyme linked immunosorbent assay, which is a technique to detect the presence of antigens in biological samples. And ELISA, like other types of immunoassays, relies on antibodies to detect a target antigen using highly specific antigen antibody interactions. Okay, so let's see the next question. Name one enzyme commonly used in ELISA. So, so the correct answer is horseradish peroxidase or alkaline phosphatase. Let's see the next question. What is the first step in blotting technique? The correct answer is a separation of molecules based on their size and charge is the first step in blotting techniques. Let's see the next question. Explain the functions of PCR. So PCR is used to amplify the gene of interest. Okay, that means target gene can be amplified by using PCR, and it is used for the construction of RAPD maps, and also used for the detection of transgene in an organism. That is PCR. Let's see the next category of questions. They are matching type. Here you have to match the list 1 with list 2. So I'm going to give you the question first. Yes, Southern blotting. Blotting techniques are given. Southern blotting is used to detect the DNA. Okay, so Northern blotting is used to detect the RNA. Western blotting is used to detect the protein, as well as Eastern blotting is used to detect the glycoprotein. So the correct answer is A, B, C, D is the correct one. Let's see the next question. Here also blotting techniques are given. You have to match correctly. Southern blotting. So, which is proposed by Edwin Southern. Okay. Let's see the next one. Northern blotting. Northern blotting was given by the scientists James Alvin, David Kemp, and George Stark. So, next one is Western blotting. Western blotting is given by the scientist Neil Burnett. Okay, let's see the next one. Eastern blotting, which is used to detect the post-transcriptional modifications of a protein. So for this, the correct answer is CBDA is the correct one. Okay, so let's see the next question. Here you have to match the list 1 with list 2. Their ELISA types are given in the list 1. Okay, let's see the matching. Direct ELISA. Okay, so direct ELISA is correctly matched to detection of antigen or antibody. Indirect ELISA, so which is correctly matched to detection of antibody. Sandwich ELISA, which is used to detect highly efficient in a sample in sample antigen detection. Okay, so let's see the next one. Competitive ELISA, which is used to inhibition ELISA. So that is the correct one. For this, the correct answer BADC is the correct answer. Let's see the next question. Next category of questions, they are belonging to true or false category. You have to tell whether the statement is true or false. Let's see the first statement. Southern blotting is used to detect RNA in the sample. No, this statement is false statement. Why? Because it is used to detect the DNA in the sample. Next one, Northern blotting is used to detect specific RNA sequences. Yes, this is the correct one. Northern blotting is used to detect the specific RNA sequences. Western blotting detects proteins using antibodies. Yes, this is also a correct statement. Okay, true statement. Next question, agarose gel electrophoresis may be used to resolve respectively 20 kb to 2000 kb DNA. So this is the false statement, because up to 100 kb it will detect, otherwise 2000 kb is the larger one. It can't resolve that much of DNA. Okay, let's see the next question. Blotting is highly sensitive and a specific method. So for this, the correct answer is true. Yes, which is a sensitive and a specific method. Next question. So the substrate in ELISA reacts with enzyme to produce measurable color change. Yes. So this one is also a true statement. Okay. So this is all about the questions. Now I'm going to give you the questions with answer. Okay, students, thank you. Thank you for watching.